So my topic is going to be on the effects of volatile anesthetics on the fetal brain. This session is kind of unique in that mine is predominantly basic science driven, whereas the other two topics are more with a clinical perspective. So first and foremost, I'd like to disclose that I have no conflicts of interest, and all the research reported here has been funded by foundation and federal sources. So historically, one of the main concerns has been the effects of anesthetic agents on the fetus, and primarily it translates to teratogenicity in the first trimester. So even before I go ahead with my talk, I'd like to have a show of hands. How many of you in the audience firmly believe that anesthetic agents given in the first trimester are teratogenic? I see one hand, maybe 2, 3, 4. Okay, excellent. So what I'd like to do is to just concise what's out there. Most of the studies have been done in animal models. There's only a very few human studies, and that's predominantly human occupational exposure studies. So what the basic science studies say that halogenated agents are probably okay. And the only problem is with the use of nitrous oxide, where it's been shown to increase the incidence of fetal resorptions. And as you mentioned before, all the clinical studies have predominantly looked at subclinical exposure to anesthetic agents in the setting of the operating room. And they've typically looked at dental nurses exposed to nitrous oxide and stuff like that. And again, the evidence is actually pretty good for nitrous oxide. It's been shown to increase the incidence of first trimester abortion. However, it's still not been proven that nitrous oxide is teratogenic. So with first trimester pretty much excluded as a period of vulnerability, is there any reason why we must be concerned? Well, there are at least many studies actually, both in rat models as well as in primates, suggesting that anesthetic agents, when given during the third trimester and beyond, cause apoptotic neurodegeneration of the fetal brain and results in learning disabilities and behavioral abnormalities as they grow up. So one of the first studies that actually looked at clinical exposure to 1 Mac isoflurane during the third trimester in rats actually revealed no evidence of histopathological abnormalities or any behavioral changes in the offspring. And this was followed by a really good study which was published in Anesthesiology in 2009, where they looked at almost 497 patients. This was actually a birth cohort from the Olmsted County in Rochester, Minnesota. And what they looked at was the incidence of learning disabilities in kids born through cesarean delivery and exposed to general anesthesia or a neuraxial technique. And they concluded that the incidence was same both in vaginal delivery and cesarean deliveries with the general anesthetic. So it looks like the data doesn't hold up, and it seems that third trimester, especially in the setting of a cesarean delivery, seems to be minimal risk to the fetus. So having excluded the first and the third trimester as possible periods of vulnerability, the question arises, is the second trimester vulnerable? And as you would probably listen to the next talk to follow, non-obstetric surgery, fetal interventions, which Dr. Upadhyay talked about. The problem with these cases specifically is that they are of longer duration. It can take anywhere between 2 to 3 hours. And as Dr. Upadhyay mentioned before, most of these procedures require a higher MAC, because if you're going to be doing a procedure close to the uterus, you're going to be stimulating the uterus and inducing preterm labor, so tocolysis is paramount. And one of the ways of achieving tocolysis is by increasing your concentration of volatile anesthetics. So they typically need a MAC of anywhere between 1.5 and 2 or maybe 2.5. But what's interesting is that the fetal brain, especially during the second trimester, is at a peculiar stage of neurodevelopment. So I've been talking about these stages of neurodevelopment. So let's take a quick look at what really happens. So early brain development is a series of precisely orchestrated steps. Neurons are generated from neural stem cells. They migrate really long distances to preordained locations where they make synaptic connections, and finally, they form circuits. So what's interesting is that most of the studies have focused on synaptogenesis, which is a stage of brain growth spurt. At least in humans, it occurs towards the end of third trimester, but predominantly it's a postnatal phenomenon. But what we are interested in is the effects of anesthetics on the stages that precede this in utero. And that would include neurogenesis, which is generation of functional neurons from neural stem cells, and neuronal migration. And again, what's important here is that both of these processes are mediated by GABA and glutamate, exactly the same receptor mechanisms through which anesthetic agents work. So the question is, would a prolonged unphysiological stimulation of the fetal brain with GABA and glutamatic drugs derail fetal brain development? So that's a big question. And right now, it's a big black box when you've just started to open it. So research from a lab which was published recently, we looked at the effects of anesthetic agents on neurogenesis, and specifically, we looked at neural stem cell proliferation. And this study was the first to actually show that there was a decrease in neural stem cell proliferation in a dose-dependent manner with isoflurane. So the way we did it was in vitro with neural stem cells harvested from rat brains and exposed them to varying concentrations of isoflurane. Along the x-axis, you see this term EDU, which is a thymidine analog which accurately describes the proliferation index. And all the data here are expressed as percentage of controls. And you can see that at 0.7% isoflurane, there's no decrease in proliferation. But with 1.4 and with 2.8%, you can see up to 20 to 30% decrease in neurogenesis. So having established this, we asked the question whether the similar changes happen in vivo. And to study that, what we did was we randomized pregnant rats at embryonic day 14, which is second trimester in humans, to receive either a control treatment with 100% oxygen for 4 hours or receive isoflurane 1.4% for 4 hours. And then they were recovered, allowed to deliver on embryonic day 22, which happens to be the term in rats. And after that, what we did was we did behavioral testing on these adult offspring, starting on postnatal day 60, which is early adulthood in humans. And following behavioral testing, we sectioned the rat brains and looked at histopathological changes in the brain. This is a timeline of the behavioral testing we did, and I'd like to draw your attention to two important tasks here. So one is the elevated plus maze, and the other is the radial R maze task. And I'm just going to highlight what these studies actually measure. So the elevated plus maze is actually a maze which is shaped like a cross, and it's raised from the ground, and one pair of opposing arms has walls in it. So these are called the closed arms, and the other arms are actually open to air, called the open arms. And what it does is it actually pits the tendency of rats to explore against its tendency to actually stay in a secure location. So typically, rats would stay in the closed spaces between the walls. The next is the radial R maze, which is a test, a really sensitive and well-validated test for hippocampally mediated spatial memory. A typical example for us would be when we get down from the elevators, we really don't know where we are. It takes about a few microseconds for us to realize which way to go. We look at visual cues, shops, paintings on the wall, and then we decide where to go. And that's exactly the same phenomenon in rats. It's evolutionarily conserved. And it consists of 12 radiating arms from a central maze. And the idea here is to get the rat to explore all 12 arms using visual cues which are put up on the wall. Rats are colorblind, so these geometric designs need to be in black and white. And what we look at is the time required for the rats to complete the maze task. That is to explore all 12 arms. And we also document what's called errors of commission, when they move down the same arm again, and errors of omission is when they fail to explore the arm. And after that, we sectioned the animal brains into 50 micrometer coronal sections, stained them with crystal violet, and then we looked at histopathological changes using the optical fractionator principle. And we specifically looked at the hippocampus, and obviously CA1 and the dentate gyrus are components of the hippocampus. And this is the stereology setup, consists of a Nikon E2000 microscope connected to a CCD camera, and is connected to a software called the Stereo Investigator, which kind of gives you accurate counts of neurons and other cell bodies in the brain sections we're studying. And these are the sample images. The first image on the left is a low magnification image of the hippocampus. And the V-shaped image on top is the CA1 area, and the arrow at the bottom is the dentate gyrus. And the images on the right side are high-power images of the same structures. Statistical analysis was done with students' t-test and with hierarchical linear models for behavioral testing, and statistical significance was accorded to p-value of less than 0.05. Take a close look at the results. Anesthesia was well tolerated by the pregnant dams, as evidenced by hemodynamic and metabolic stability. We do monitor blood pressure, we do monitor glucose, and we actually do a typical anesthetic in rats. We do monitor everything what we would do for a human patient. There were no differences in maternal weights between the two groups, both on embryonic day 14 when they were exposed to anesthesia and the day before term. And there were no differences in the offspring between the control and anesthesia groups, both in terms of the litter size and also the weights of adult male offspring. So the task which I mentioned before, the elevated plasmase for anxiety-related behavior, what we found was that anesthesia-exposed animals preferred open arms. That is, they made more open arm entries and also spent a long time in the open arms, which indicates that they have less anxiety as adults. And when it comes to the radial arm maze, rats exposed to anesthesia in utero took a really long time to complete the radial arm maze task and also made a number of omission errors in the task, indicating that anesthesia exposure in utero would impair hippocampally mediated spatial working memory. And this is important because this was the first study which actually showed that a single exposure to anesthetic at a critical period of neurodevelopment causes behavioral abnormalities which are temporally distant from the time of exposure. So just imagine getting exposed to anesthesia in the second trimester, just one single episode, and there is impaired neural development for the rest of your life. So with the stereology, where we looked at histopathological changes, there were no differences in the cell numbers in the CA1 region. But what we found was that there was a 10% decline in the total number of cells in the dentate gyrus, which is really critical for forming new memories. And this is true for human beings as well. And so what I'd like to conclude is by saying that in utero exposure to isoflurane causes a persistent region-specific cell loss in the hippocampus and also causes behavioral abnormalities. The causative mechanisms are currently unclear. We are still kind of exploring what the possibilities are. And obviously, rats are rats, humans are humans, but what we have tried to show here is to establish the proof of principle. And so it has to be confirmed in private models and preferably in humans. And this will have significant implications in the choice of anesthetic agent for non-obstetric surgery. And there have been a couple of papers which have compared isoflurane to other volatile anesthetics like Sevo and DES. And it looks like there's not much of a difference between volatile anesthetics with regards to neurotoxicity. Propofol is a promising option. My lab is still working with propofol, and we do have in vitro data suggesting that they do not impair neurogenesis. Obviously, we have to look at other findings such as behavioral abnormalities, and until then, I cannot say for sure whether propofol would be the way to go. So to summarize my talk, in the first trimester, no teratogenic effects with the possible exception of nitrous oxide. So you're pretty safe. And in the third trimester, as marked by green, no adverse effects on the fetus in the setting of a cesarean delivery. But what's probably going to be a problem zone would be the second trimester, because as you mentioned, a lot of surgeries get done in the second trimester these days, and it's a stage of active neurogenesis and neural migration in the fetal brain. And we have shown that anesthetic agents do decrease neurogenesis and also decrease the cell number and cause behavioral deficits. So I'd like to conclude my talk by acknowledging a few people here. My mentors, Gregory Crosby, Deb Cully, and Lawrence Sen was my clinical mentor. I'm sure a few people here in the audience might recognize him. And my collaborators, Pradeep Bide and Mark Baxter, and my research techs, Bess, Emily, Laura, and Deirdre. And I'd like to conclude my talk by invoking this Hippocratic precept, primum non nocere. First, do no harm. Thank you.
And actually, it is quite interesting to hear all this in the experimental model, but how it is going to translate in the clinical setting and the long-term effect on a child who has been born, been given anesthetic agent during the second trimester needs to be seen.
So you bring up a really important point. And the problem with human studies especially is that there are a lot of confounding variables. And most of the time, learning disabilities are picked up later in life. It's not like you detect learning abnormalities when the kid is 3 years. So it takes 7, 8 years, and no one makes the connection between what happened in utero to what's actually happening at that point in time. So it's really important, and we're still looking at ways how this could be picked up earlier. And as I said, we are just trying to establish the proof of principle, because we have typically taken anesthetic agents for granted. We've always looked at macroscopic findings, looking at heart rate, looking at effects of anesthetics on the APGARs. We have never looked at anything beyond that at a microscopic level. And only the past 5 or 6 years that a lot of studies have come out indicating that anesthetic agents are probably toxic to the brain, and we probably need to be careful in what we're doing.
Thank you.
Thank you.